particular lecture, I shall be discussing the wall aspects of bacterial reproductive structure or the endospore, the different components and their function. After which I shall be discussing the different steps by which bacterial cell wall is synthesized or the biosynthesis of cell wall, and then finally I shall conclude with the different ways of bacterial cell wall degradation, which include the enzymes from bacteria or bacteriophage and also the action of different secondary metabolites like the antibiotics and their mode of action. Now, you can see in this particular slide that how a bacterial endospore is formed. This is a bacterial cell wall, and this particular cell wall is showing some invagination, and you can see from stage 0 to stage 7 the different ways by which the endospore is formed. As it is developing, there will be an invagination and then finally formation of the pre-spore and then gradual thickening of the pre-spore, and the protoplast is getting inside, and finally the endospore is produced and it is getting separated from the empty cell to give rise to this particular structure. So, what is this structure? This is a complete endospore which is a very thick wall which include the exosporium. The exosporium, spore coat, cortex and the core wall are the primary layers, and in the exosporium you will mainly have protein and lipid. Protein may constitute up to 52% and lipid may be around 18%, which include the cardiolipin, which include fatty acids in the order of say 16 to 18 carbon, and also the protein will have different hydrophobic amino acids, and finally after the exosporium there is the spore coat. And this spore coat actually is also formed of hydrophobic amino acid which is protecting the cell wall from other invasion, and this particular spore coat is also protecting the bacterial endospore from lysozyme action. Then finally we have the cortex, which is nothing but a peptidoglycan with dipicolinic acid and calcium, and finally we have the core wall which is just bordering the protoplast, and the core wall you know is formed of the small acid soluble protein SASP, and the innermost region you can see this is the genophore or the nucleoid region, and this particular endospore will also have ribosomes, but you know the metabolic activity of this particular structure is very little because it has to survive the unfavorable condition when the bacteria is not getting adequate food or water.
Sir, I have a question. What is the utility of calcium in the cortex of the endospore?
As I said that the cortex contains dipicolinic acid and calcium. So, calcium will bind with this acid to produce an acid salt, and this provides stability and thereby also provide rigidity to the cell wall and protect it from other bacteriophage enzymes. Peptidoglycan you know is the main component of bacterial cell wall. It is present both in gram positive as well as in gram negative cell wall, though we say that in gram positive cell wall it constitutes up to 85%. Now, in this particular slide you can see that how this peptidoglycan is produced in a typical gram positive bacteria called Staphylococcus aureus. Now, there are two major components. One is the production of the sugar residue and the other one is the incorporation of additional component in the form of teichoic acid. So, broadly a particular cell wall will have two major steps of biosynthesis. One is the synthesis of the peptidoglycan component in the form of sugar and protein, and the other one is the formation of the secondary acid components of teichoic acid if it is gram positive bacteria, because in gram negative bacteria you do not have teichoic acid. So, here primarily this particular component is the most important component that we see in gram positive cell wall. You can see here that this particular layer is the membrane. So, it is the membrane which is producing all those enzymes which are required for the biosynthesis, and you can see that initially there is the UTP sugar by MAR-A will be attached to ennoyl phosphate, and this ennoyl phosphate residue will finally produce the second step which is the MAR-NAC residue, and then there will be a series of event. Actually this event will be controlled by MAR-C, then MAR-D, then MAR-E and MAR-F. and this particular steps actually indicate attachment of the amino acid to the sugar residue. So, this Marnac sugar residue will be having the amino acids of alanine followed by glutamine followed by lysine, and then the lysine again here is followed by alanine. So, this particular sugar residue attached by the amino acid, or you can see 1, 2, 3, 4, 5 pentapeptide linkage constitute the most important component of the peptidoglycan, meaning sugar and amino acid. And the most important step being the initial step, the MAR-A step, which produces the typical enol pyruvyl activity, and then there will be the reduction at the MAR-B step followed by continuous attachment of the peptide. Now, one important thing is that the MAR-E, which is the actually the fifth step, in gram negative there will be diaminopimelic acid. So, attachment of diaminopimelic acid is there if it is gram negative, but if it is gram positive the diaminopimelic acid is not there. So, there will be the lysine attachment as you have seen here. So, there is the variation between gram positive and gram negative, but what happens after that? That the MAR-F will actually result in the completion of the UTB NAM pentapeptide unit, and this unit you can see is produced in the inner membrane of the bacterial cell wall, which will now be transported, and this transportation is being done by the MRAY. The MRAY is actually getting attached to the undecaprynyl phosphate, which will produce the lipid 1, and then finally the MAR-G will transfer it to lipid 2. But mind you, this lipid 2 formation is not the end of the step. By some mechanism, this particular unit will be transported outside and they will produce the basic unit of the peptidoglycan. So, peptidoglycan biosynthesis starts in the inner part of the membrane and then it comes out, and then gradually it polymerizes, producing the entire cell wall. You can see the formation of this sugar protein residue is being formed inside and it is transported outside by MRAY, and finally the MAR-G is involved in the formation of the lipid 2. And then finally when it is transported out, there are two important components, namely the TG and TP, which are the two components of the penicillin binding protein or PBP, which is involved in the transfer of this cell wall residue outside the plasma membrane into the outer surface of the plasma membrane, producing the cell wall. So, this is the most important step where the unit is produced inside and is transported outside. There will be the intracellular changes. You can see here the formation of the amino sugar 1 and 2, then attachment of the amino acid residue producing the pentapeptide, and you know the most important step being alanyl alanine formation by the enzyme synthetase or the transfer of L-alanine to D-alanine, and then finally alanine binding with another alanine producing the alanine synthesis, which is also targeted by some antibiotics. And the next is the formation of the sugar residue, and this is the amino acid residue, this is the step 2, and the third one is the transfer of this particular unit outside the particular cell membrane, producing the cell wall outside. And this peptidoglycan, you know, is the very important protective structure in gram positive, and in gram negative it is not there, but it is replaced by the LPS layer. This component is called the lipid teichoic acid and also the wall teichoic acid. In this particular structure, you can see the lipid teichoic acid is formed of phosphate and it is also having glycerol and ribitol residue. So, ribitol residue attached to phosphate producing a long chain will produce the lipid teichoic acid. Sometimes the ribitol may be replaced by glycerol. Now, coming to the position of the wall and the lipid teichoic acid, you can see this is the typical lipid teichoic acid, a straight chain where it is actually having a similar type of compound, but in wall teichoic acid you have the different residues. You can see in this particular slide the different steps are involved. there is always a backbone. I told you the backbone is undecaprynyl and the phosphate which is binding with the sugar residue. So, sugar residue will be gradually getting attached, namely the glucose and the mannose residue, followed by the glycerol, and then this 2-glycerol residue followed by the long ribitol residue. And this particular step you can see is controlled by the tag protein, the tag O, the tag A, the tag B, and then the tar F and tar K will complete the formation of this particular glycerol sugar residue, and finally it is the tar L which will continue its activity until it produces a long chain of the waltecoic acid. And this particular step of waltecoic acid formation is actually very important in the completion of the gram positive cell wall biosynthesis.
Sir, I have a question.
Yeah.
Which is more effective in LTA and WTA?
You see, LTA and WTA both are providing rigidity to the bacterial cell wall, but in case of WTA there will be the attachment of the phosphate, there will be the attachment of the ribitol or glycerol both, and at the same time WTA is projected outward as receptor. So, in that way you can say that WTA may be more effective or functionally more dynamic than LTA. Clear?
Yes, sir.
So, the transfer of glycosyl group is a very important step, and this glycosyl transferase activity you can see will lead to the formation of the first unit, which is lipid 2, and I told you in this particular form it comes out of the membrane, then it gets into the lipid 4, and then finally n number of residue will join to produce the long cell wall or the long peptidoglycan unit or the peptidoglycan layer of the bacterial cell wall. So, in a bacterial cell wall we have two major activity. This is transglycosylation and the other one is transpeptidation, which occur independently. You can see in this slide, this is the site of transglycosylation and this is the site of transpeptidation. These are the two very important activities. Now, naturally these activities are being controlled by a unique protein, the penicillin binding protein or the PBP, and this PBP you can see is actually having two major sites, the TP and the GT, and they are joined by a linker. And you know the GT is actually attaching the glycosyl residue, whereas the TP is attaching the protein residue, and in this way the TP and GT will work side by side and thereby they will produce the unique bacterial cell wall component.
So,
and yes.
So, you're saying that both of them are occurring, transglycosylation and transpeptidation are occurring simultaneously.
Okay.
So, but does transglycosylation come before transpeptidation?
You see, transglycosylation and transpeptidation apparently occur simultaneously, but considering an experiment where they have used cell-free system, there they have seen that transglycosylation can occur independently. So, we can say that the decisive step is transglycosylation, which is followed by the transpeptidation, and definitely they are at a later stage taking place simultaneously. Now, coming to a very important structure, which is the multifunctional glycosyl transferase or MGT, which is functional in certain bacteria. This particular peptidoglycan synthesis in E. coli or in Micrococcus, their MGT is functional, and this MGT is bringing about the glycosyl transferase activity. So, it is an additional protein which is actually bringing upon the glycosyl residue formation in certain gram negative bacteria. We come to the different ways of bacterial cell wall degradation. Here we will be discussing the different aspects of lysozyme that degrades bacterial cell wall. Lysozyme may be either produced from our body or it may be viral in origin. Then we will be following it with the different antibiotics that degrade bacterial cell wall, and finally we will conclude with the different bacterial enzyme that is bringing upon autodigestion of the bacterial cell wall. So, the starting molecule is the lysozyme. The lysozyme is a unique protein or an enzyme which is produced from the tears. It is present in the saliva, it is also present in the mother's milk, and this lysozyme is mammalian in origin and it is degrading bacterial cell wall. And the lysozyme, as you know, is actually breaking upon the beta 1,4 glycoside linkage between the sugar residue. So, when the beta 1,4 glycoside linkage is being cleaved, the sugar residue is getting broken up. And when we consider that lysozyme is only present in human, it is wrong. Lysozyme is also present in bacteriophage. This phage actually is degrading bacterial cell wall, and you can see the lysis of bacterial cell wall by phage enzyme. It is actually having two definite mechanism. One is the one gene lysis or one gene system, and the second one is the two gene system. Suppose this is the enzyme, it is binding with the MRAY. You know, MRAY is actually the translocase protein which is translocating the peptidoglycan unit outside the membrane. So, it is being blocked, and as a result of which the cell wall gets degraded. The second one is a two gene lysis where there are two different components. One is the choline and the other one is the endolysine component. The choline actually will bind with the cell wall component of the bacterial cell wall, degrading it or causing distortion of the membrane, and finally the endolysine will get in and will break the different parts of the bacterial cell wall. Now, definitely there are different the bacterial cell wall component called secondary cell wall polymers, and these secondary cell wall polymers are present in the different parts of the bacterial peptidoglycan, and they are being attacked by different bacteriophage. This bacteriophage is producing different groups of enzymes like the alanine amidase, the muramidase, the glucosaminidase, the endopeptidase, the glutamate endopeptidase, and finally the transglycosylase. These groups of enzymes are being produced by the bacteriophage, and it will bind with the different components of the bacterial cell wall, and these components include the peptide linkage, it will also include the different part of the sugar residue, and ultimately it acts as a diverse enzyme, or I should say it is a versatile enzyme which will definitely bring about the destruction of the bacterial cell wall.
Sir, I have a question.
Yeah.
What is the exact role of the secondary cell wall polymers?
You can see here the secondary cell wall polymer is present mostly in gram positive bacterial cell wall, and these polymers are actually imparting rigidity. It is imparting rigidity to the peptidoglycan, giving it a definite shape and structure, and at the same time it is acting as a definite receptor for this phage enzyme, and which you know is there in the form of choline. So, this choline receptor is actually being brought about by the SCWP, and thereby the bacterial cell wall is getting degraded by the phage enzyme. There are different forms of antibiotics which are mainly classified into two types. One is the cell wall inhibiting agents and the other one is the beta lactamase inhibitor, though here I am not considering those antibiotics which are protein synthesis inhibitor. I am particularly telling you about the different cell wall inhibiting antibiotics which include the beta lactam and the other antibiotics. The beta lactams, most of them are including the penicillin, the cephalosporin, the carbapenem and the monobactam. Now, these penicillins include amoxicillin, ampicillin, penicillin G, penicillin V, etc. And you know these penicillins are mostly now not in use because the bacteria have become resistant to this particular penicillin group of antibiotics because they produce an enzyme called beta lactamase which destroy the penicillin molecule and will not allowing the penicillin to act on the bacterial cell wall, mainly the peptidyl linkage, and thereby the penicillin is rendered ineffective. Then we have the cephalosporin. Mind you, the cephalosporin is a group of antibiotic having the cepham ring which is divided into first, second, third and fourth generation. The first generation like cefadroxil or cephalaxine, the second generation being chlor, the third generation being cefixime, the fourth generation being cefepime. Now, why these molecules are developed? Because you know, if the first generation is having limited utilization, then we go for the second molecule, and this development primarily is by changing the side chain of the particular cephalexin molecule. And we also have the carbapenem in the form of imipenem or meropenem, and we also have the monobactam, that is the aztreonam. Now, one important thing is the cephalosporin group of antibiotic, and this penicillin, you know, is different from cephalosporin. And how this cephalosporin is active? They are also inhibiting the cell wall biosynthesis of different bacteria, and thereby they are preventing the peptidoglycan formation. Now, coming to the next group, which is the general group of antibiotics called carbapenem. Now, carbapenem molecule is divided, it is having different molecules like the imipenem, the meropenem. You can apply it anywhere. Now, this particular imipenem or meropenem will penetrate inside the cell wall, and thereby it will be active over the peptidoglycan. Though they are both carbapenem, imipenem may be more active over meropenem. Imipenem and meropenem molecule is being provided in this particular cell wall. Now, if it is a particular 2-layered gram-negative bacteria, then there will be first level entry and then second level entry. Now, this entry successfully is made by imipenem, but not by meropenem. Meropenem will be eliminated by some mechanism, which is the efflux system of MEX-B or MEX-A, which is taking it out into the outer membrane, and finally the OPRM will be pulling it out or putting it out of the cell wall. So, before the molecule is active, then the molecule is being thrown away of the bacterial cell wall. So, look how smart is the bacterial system in order to cater resistance to costly antibiotics like meropenem. Now, coming to a group of antibiotics called monobactam or aztreonam. This is the aztreonam antibiotic, and this particular monobactams actually will also be effective on bacterial cell wall, and they mostly will be acting with the monocyclic beta-lactam nucleus, or which is getting attached to the bacterial cell wall. And those cases where the bacteria is capable of producing beta-lactamase, this beta-lactamase will actually destroy penicillin, but they cannot destroy monobactam. So, aztreonam will be functional in those cases where there is beta-lactamase production. This particular monobactam may be a costly antibiotic. So, in order to reduce the toxicity, in order to have low cost antibiotic, people will mix cephalosporin or penicillin group of antibiotic with the beta-lactamase inhibitor. So, thereby these days plenty of antibiotics are combinational therapy, where they will combine the penicillin or cephalosporin with the beta-lactamase in the form of clavulanic acid. Now, the other group of antibiotic is the vancomycin. This vancomycin is actually unique. It is binding the alanine-alanine dipeptide linkage. Suppose this is the NAG-NAM chain, and they are being linked by the tetrapeptide, and this tetrapeptide, the terminal part is alanine-alanine, and this alanine-alanine biosynthesis is being inhibited by the vancomycin group. So, if that particular vancomycin antibiotic is applied, they will bind with the alanine-alanine residue and will bring about the breaking of the tetrapeptide linkage, and thereby will disrupt the bacterial cell wall. This particular antibiotic was discovered in 2001, and in 2003 there was resistance to this antibiotic. How this resistance was brought about? This vancomycin resistant group actually will not allow the free alanine to bind with the particular vancomycin molecule. So, this particular cell wall cross linking enzyme will be active because the vancomycin is not binding with the alanine residue. So, the cross linkage is completed. The third and the fourth amino acid will be combining with the pentaglycine linkage, and the structure of the cell wall is maintained. So, vancomycin, though it is a costly antibiotic, it is not really functional in gram-negative bacteria. Why? Because in gram-negative bacteria, the peptidoglycan component is very little, and that is the reason why gram-negative bacteria are well protected to vancomycin, though it is a very costly antibiotic. Beta-lactam ring is the central ring of a particular antibiotic, and this beta-lactam ring is being cleaved by an enzyme beta-lactamase, and for this many reasons that this group of penicillins, the cephalosporins, the vancomycins, they are of no use, and those particular bacteria can be killed by another group of antibiotic which is called beta-lactamase inhibitor, meaning before that bacteria produce that enzyme beta-lactamase, it is being blocked by this beta-lactamase inhibitor, like the clavulanic acid, and will produce an acyl enzyme complex, and thereby this beta-lactamase cannot bind with the active site of the molecule penicillin. So, what happens is that particular penicillin will be active, and it can go in and it can cause breakage to that particular alanine linkage, and thereby the cell wall will be cleaved. These days only penicillin with beta-lactamase inhibitor is not adequate. So, these days doctors, they do not take any risk. They get into the third or fourth generation cephalosporin, and they will mix it with the beta-lactamase inhibitor, and thereby these drugs are very effective in the present day. But you really never know in future what is stored for us, because you know right at the present moment we are firing a cannon against a mosquito, but you never know if that mosquito becomes a demon at a later stage, so the cannon will not work. That means for petty infection you are using costly antibiotic, for the time being we are being saved, but in later time you never know what is stored for us. Now, coming to the last step of bacterial cell wall degradation, where the bacteria itself is degrading the different components. One important step is the action of amidase. You know, bacterial cell wall may have amides, and these amides are being acted upon by amidase, which will generate the amino acid, and the extra amino group will be released. In the same way, there may be the acetyl glucosaminidase, which is produced by the bacteria, where the N-acetyl beta glucosaminidase will act upon the glycoside linkage, breaking the sugar unit and disintegrating the bacterial cell wall. We have started with the endospore wall component, then we have discussed the different ways of bacterial cell wall biosynthesis, and then we have got into the different ways of bacterial cell wall degradation, degradation by lysozyme, degradation by antibiotics, and also autodigestion by bacterial enzyme. Thank you.